topic of my presentation is the review of QSAR nanoparticles with its toxicology. The contents of the presentation are as follows. The first will be the introduction of the quantitative structure-activity relationship, followed by the different models of QSAR which are being used in bioinformatics. Then we'll be having an idea of what nanoscience is and what nanoscale is and how interdisciplinary the field is. Then how these nanomolecules interact with the biocellular mechanism. We'll have a look into it. Then study how this nanobio interaction is also causing some toxic effects in the cellular environment. Then the integration of nanoinformatics with drug design and what are the different tools that are presently available. Different computational modeling techniques are used in QSAR. And finally, we would like to conclude the presentation with the toxicity effects of nanomaterials. So, to get a brief idea of what QSAR is, QSAR stands for Quantitative Structure Activity Relationship. So QSAR is a computational modeling method that is used to identify the relationship between the structural properties and the chemical compounds and the biological activities. And what you can see in this diagram here is the different types of chemical activity one can do. So this activity, how we can model using the QSAR software. This is how we can develop new compounds with biological activity. This we'll be knowing in depth. So there are two purposes of using QSAR. This is mostly used for predictive modeling. So the prediction of endpoint values for understanding the chemical for data gap filling. And the other one is a mechanistic approach in which a physicochemical and a mechanistic interpretation of the structures and the response are studied. QSAR has different modules. Here is a list of different modules under which the QSAR is being widely used. So first is a structure activity. In the structural activity, the chemical descriptors and the molecular properties, these are studied with respect to the biological activities. So these can correlate the structural feature of molecules with the help of biological activities. So these models are very widely used in drug designing and molecular biology. Similarly, as structure activity, here there is an activity that is important, but in QSPR, it is a property which is highlighted. The properties such as the physical, chemical, and biological aspect of this bioentity are studied with respect to the structural features. And then QSAR ADME. This is in the field of pharmacodynamics. So in this, the adsorption of the molecules, the distribution, metabolism, and elimination of the molecules are studied in a molecular modeling approach under a certain environment. Then the structure toxicity. In structure toxicity, how this molecular structure is toxic to the environment by assessing the safety and the potential drug candidate based on the chemical structure studied. Then coming to the omics part. So after toxicity, this QSAR is also used in the omics studies dealing with the proteomics and genomics. Proteomics with the interaction with the protein entities, and genomics at the gene and the genome level. Then cheminformatics, it is interrelated with the structural activity and the structure property. This is used to study the activity of chemical compounds, and this is one of the valuable tools which is being used to discover for new drug identification and drug design. Then toxicogenomics. This is to integrate the toxicologic and the genomic data to predict the toxicity. The structure binding. Now, when there is a molecule, the molecule has different types of forces within itself. It could be different affinity molecules, and how this interaction of the molecules happens with the help of ligands, receptors, and enzymes. This is studied. And finally, the structural biology, which is based upon the interaction with the biological molecules like the protein and the nucleic acid, which forms the part of proteomics and genomics. Then what is nanoscience? Nanoscience it involves the design, manipulation, and application of structure, devices, and systems at nanoscale. Nanoscale is nothing but 1 to 100 nanometer range. So this in this range, how the materials behave and the properties behave. This is about the nanomaterial. What are the different properties of nanoscale? So different materials exhibit different wide range of properties at this level. So they exhibit novel properties. So what materials are being there as a bulk material? When you scale it down to nano range, the materials properties only changing. For example, electrical property is changed in the case of silver, copper, aluminium, iron, and zinc. So this bulk material they have low conductivity, but at nano they show a very high conductivity. The mechanical property of nanomaterial can be seen in this diagram of a SEM graph. In this SEM graph, what we can see is these atoms. These atoms, this is of the diameter range of 289 nanometers. But when the size of this diameter is reduced to 59 nanometers, what is happening is this mechanical property, hardness of 30 megapascal is being improved to 270 megapascal. So this is how it is creating a vast impact in the nano range. Then optical property is increase in transparency. Now you can also see in this diagram how the transparency is being increased from going from bulk material to decreasing the diameter and pore size of the material. And then in chemical property, it has increased catalytic property and reaction rate. Magnetic property, it shows more magnetism when compared to the bulk material. Now, at nanoscale, bulk materials are characterized by the properties of composition, solubility, melting point, boiling point, vapor pressure, and pH. But with the decrease in size, characterizing of nanoparticle changes. For example, the size, shape. So you have different characterization techniques which are being used in order to characterize these materials. So you have melting point, surface area, wettability, surface tension, band gap, pore size. So these are being done by using advanced characterization techniques like SEM, XRD, AFM, NMR, etc. So at this nanoscale, one of the important feature is the surface to volume phenomena, which takes place. So in this diagram, what we can see is a cube. So this cube has one side, and its surface area is a square. That is one square into its six sides. So 6a square. So 6 into 1 square. So now when the surface area to volume, when you consider it of two sides, then it comes up to 24. So over here what we can see is from 6 it is going to 24, and it is having a surface of 54 dimensions in which the material can exhibit its property. So the different material examples are being shown in this figure. Like copper nanomaterial, initially it shows transparency nature as its molarity increases. Then this is platinum nanoparticles. This is being used as a catalyst application to increase the reaction rate in different industries. And this is the aluminium nanoparticle, which you can see here. This nanoparticle is it is at room temperature stable, but once it is at nanoscale, it becomes combustible. Next, what you can see here is normal room temperature occurs in solid form, but at nano range it is transparent in nature, and it is in other form, liquid form. And silicon, it appears to be a conductor, but when it is in nano range, it is an insulator. So nanoscience explores about the phenomena and manipulation at the nanoscale. So this field intersects with various scientific and engineering principles. So these are the various fields in which nanoscience is being studied vastly. First, about nanomaterials, it is used in the preparation of nanoparticles by chemical reduction methods or any method. These nanoparticles are being synthesized in various research labs for different application purposes. And these nanocomposites are nothing but molecules with two or different materials. Like For example, silicon carbide. So you have silicon particles and carbide particles which are incorporated to form the composite materials. Then we have these devices. These devices, for example, they are the application part of it is mostly seen in integrated circuits, which are being integrated circuits of laptops. So how they are done is by the process of fabrication, and nano generators also are being developed in which the energy gap is being modulated, and lab on chip devices. This is mostly in respect, it is a device but with respect to medicinal use, like for example this glucose testing and all can be done with the help of by modulating the devices and the medicinal part together. Then electronics, it is used in the application of quantum dots. In quantum dots, how the light plays a key important role in which, for example, a lipid nanomolecule, when it is exposed to light, what happens is it shrinks and it unshrinks. So that phenomena can be studied. And in different transistor and synchronic approaches. In the case of characterization, as I mentioned below, there are large number of advanced instruments that are being used to study and image these molecules. Uh, Coming to the medicinal aspect of nanoscience, it is widely used in drug delivery for nanocarrier molecules in order to transport the drug uh, to the local area of the target, and it has various applications in nanobiotechnology. Then this nanotechnology also is being used in chemical and biological sensors to detect various chemical cues and molecular cues. It is used in robotics in order for the manipulation. Like nanoscale is nothing but manipulation of matter at a size range of 10 to the power of minus 1. Then it is having different tribological study nanobio interaction. I have divided. This is a diagram that is representing the engineered nanomaterial, and this is a nanomaterial, and this is with respect to the physical chemical phenomena. This is with respect to the cellular aspect of it. So this engineered nanomaterial, it can exhibit at its surface property. This is the surface of this nanomaterial. So it can exhibit n number of properties at its surface level. So this engineered nanomaterial at its surface level can have defects in it. It can absorb sorbate. Sorbate is nothing but a chemical cube which it can absorb. Then it can have pH. It can be pH sensitive. It can be stereo selective interaction. There can be electrostatic interaction, hydrophilic interaction. Then there could be an interaction of ligand to receptor, and reactive oxygen species, that oxygen radicals can interact with this molecule, and enzyme inhibition, and any type of property, and this property can be studied and modeled using computational modeling of using QSAR. And so now when you come to the cellular aspect of it, what happens is when this engineered nanomaterial can interact with any biological entity such as carbohydrate, siRNA, DNA, oligonucleotide, antibody, photothermal therapy, contrast agent, and peptide. And this interaction point, it forms what is called as a protein corona formation. The plane at which the engineered nanomaterial and the cellular entity forms is come together. At that point, it is called as protein corona formation. So the surface charge and the chemistry of nanomaterial influence the interaction with cell membrane, protein corona formation, and the cellular uptake. So far we have studied what nanomaterial is and what are the different types of nanomaterials and how it is used in different fields. So one aspect of this study is also the toxic impacts which these materials have in the environment, be it the cellular body or the. This diagram is a part of the cell. So the cell, as we studied in our primary classes, the cell consists of different cellular organelles. What we can see are these intermediate filaments. these are the microtubules. This is the ribosome, mitochondria, nucleus, surrounding nucleolus, and the endoplasmic reticulum. So what happens is when these nanoparticles are present, and these nanoparticles, when they try to enter into the cell, when they try to get into the cellular membrane and enter, then they can cause a haphazard type of reactions. So once when it interacts with the cytoplasm, it can form reactive oxygen species. At cellular membrane, it can damage the cellular membrane. Then it can cause reactive oxygen species, and DNA damage can cause mitochondrial dysfunction and causing cancer-like symptoms. Then it can oxidize the damage of proteins in the protein, like it can destroy the protein structure. It can destroy the mitochondria, causing the DNA damage. Then this is related to the genetic abnormalities which we can see. And in ribosomes also, the siRNA molecules can be destructed. And here the interface of lysosome. So lysosome has its own activity. So now it can cause cancer-like symptoms in this condition. So this nanoparticles, when it gets added to any of the cellular organelle at any point, it can have various types of toxic effects. So so in this slide we see how this nanoparticle. So this nanoparticle also could be a drug molecule. It could be an active pharmaceutical ingredient. It could be anything. So when this is integrated into the system, it acts as a drug. So also, like what was studied earlier in the drug delivery system or the conventional drug delivery system, were ointments, tablets, capsules, and syrups. So these were administered orally, and the type of damage was enzymatic reaction and the lower pH environment. So then the pharma companies had come up with a controlled release type of drug delivery system. So in which the release time of the drug had been monitored, such in the case of diabetes patient, where diabetes patients need to be administered drug at different intervals of time, so that their sugar is balanced. So the type of different types of controlled release examples were hydrogel, then erodible material, polymer matrix, and degradable material. So this system is fronting as a drug aggregation. So what happens is the drug is being aggregated at a single point. So due to the large size of matrices and delayed in release time, less soluble and less targeted. So this approach was being carried out, but with the help of nanomedicine based drug delivery system, nanomedicine has a targeted approach. So targeted approach, you can set the target to which organelle how much amount of drug has to be sent. So target specific transport drug to the affected areas without damaging healthy cell. Suppose there is a cancerous cell, we can target the cancerous cell with the help of molecular tubes. So the various examples are gold nanoparticles, carbon nanotubes, liposomes, and dendrimers. So now, so in this form, nanomolecules are being used to study in a drug delivery approach. Integration of nanoinformatics with drug design. So in order to study what the drug is made of the chemical structure and the different properties, we have to learn what is drug designing. So in order to learn drug designing, there are different types of molecular techniques, molecular modeling methods that are available. One is a molecular dynamic simulation, in which an environment is being modeled, and at different activity and structural aspect of it is studied. Then QSAR in silico, targeted drug delivery, as we can target the cancer causing cancerous cell, and the predictive model. Like we can with predictive models we can get an idea of what can be the outcome of the situation in a given confined environment. So this field is nothing but nanoinformatics. So to enhance the efficiency and the success of drug discovery and drug development by maintaining an accuracy in predicting the biological activity of nanodrugs, nanoinformatics is being used. So as we have seen QSAR is also used in studying the nanostructure in the form of QNAR, that is a nanostructure activity, QSPR, that is a nanostructure property, QNBRR, that is a nanostructure biological response. So in this nanostructure activity, the nanostructure material and its toxicology activity is studied. In nanostructure property, the physicochemical properties like solubility, stability, and reactivity are studied. And in nanostructure biological response like how cellular uptake and toxicity is being studied. So this is a schematic. It represents how a new drug is being investigated uh, in any for new drug investigation. So you have the hit ID, which in the hit ID you'll have a different number of cues in which it boils down to a few of them, and then the lead optimized molecule is being selected, and that is being studied. So here, these are the different optimization and the chemistry part of it. So it also uses predictive models such as QSAR and QSR3. So these are the different areas in which drug designing can be used. Then the tools which are used to study the QSAR, it is called cheminformatics, as it is the structure of the chemical information with respect to the molecular environment. So the quantitative structure activity relationship model in nanoinformatics. So this is the development of mathematical modeling to have precision medicine for the treatment that is tailored to an individual patient. So nowadays, due to our lifestyle changes, every individual is having different lifestyle disorders. So this drug designing can tailor we can tailor medicines for different diseases of lifestyle disorders on under different biological characteristics. So the different tools which we can see here is for cheminformatics study. We can use ChemDraw. In ChemDraw, we can draw structures of different chemical molecules and find out what interactions it is having. The hydrophobic, hydrophilic, the steric interaction, the bonding can be studied using cheminformatics. So there are different softwares such as Schrodinger, R, and 9. R is used for machine learning approach, and this is used for modeling for the ligand and receptor binding area. This can be studied. Then there is a QSAR world. This is used as a toolbox. This also a part of modeling and cheminformatic tool. And there are different databases to find out the different physiochemical properties uh, of the chemical. So this databases like PubChem database are there. Then this is the literature study of different toxicity of nanomaterials. So nanomaterials which are synthesized as titanium oxide and silver fluorines, it can cause internal localization, and the toxicity test for it is TEM. This is a microscope, advanced microscope. By using advanced microscopy, this intracellular localization can be visualized, and with which we can confirm that there is a toxicity being caused by this TiO2 molecule. Similarly, we have different molecules such as CNTs, carbon nanotubes, and this can be studied using the light microscopy. And the main area of interest is the metal and metal oxide nanoparticles, which cause a DNA damage, genotoxicity, cell viability, apoptosis. So these metal and metal nanoparticles, these are taken up, they are being adsorbed, and how it is interacting with the cellular organelles and causing the toxic effects. They can be tested using the micronucleus test, Comet test, lactate dehydrogenase, Alma test, and red assay test. So these all tests can be done in order to find out either the cell is alive or dead, or the DNA is causing damage at the nucleotide level, or the genotoxicity conditions and causing cancer-like conditions. So by using these tests, this can be confirmed. And it also causes oxidative stress-like condition, which is used in the study of the reactive oxygen species. So the composition of nanomaterials. So nanomaterials are classified into hard and soft. So this study had been done, and in the research articles, what was found is these hard materials are like metal oxide, and carbon based. There are soft materials like polymer, silica, quantum dots. So due to its soft nature, it can these liposome in this diagram. What we can see is the drug molecule. So this drug molecule is in a it is in a liposome. This is a dendrimer, and these are the this is a C60 molecule, single wall carbon nanotube. So in this in this uh, these these particles are produced and synthesized in the laboratories. And metal in the case of metals, silver, copper, platinum, gold, and in metal oxide, titanium oxide, cobalt oxide, iron oxide, and in carbon-based materials, fullerenes, carbon nanotubes, uh, and graphene nanoparticles, quantum dots, CDAC quantum dots, and silica particles, silicon dioxide, and in polymers, dendrimers, and lipids. So these nanomaterials are being widely synthesized, and they are used for varied applications. Now, what happens when these metals they interact with the biological aspect, that is at cellular level? So these metals, which are silver, platinum, cobalt, gold, or be it the oxide forms like the manganese iron oxide or oxides of titanium, zinc, and molybdenum, or be it silicon carbide, they can cause a DNA level damage. How this DNA level damage is done? This DNA level damage may occur at various levels. It can cause a chromosome deletion, which can cause a congenital or and genetic aberration. It can cause an epigenetic abnormality. It can cause telomere attrition, which can lead to cell senescence and aging. Then it can cause mitochondrial DNA damage, which can cause a cancer-like symptom, and it can cause DNA fragmentation. That is, SNPs and variations can be seen. Now, what happens is this healthy cell. For example, in this experiment, what they have done is these are the nanoparticles. These black dots are the nanoparticles. This part is a cancerous cell. This part is a healthy cell. So if this nanoparticles are exposed to if this part of the cell is exposed to the nanoparticle, and this cancer part is also exposed to the cell, these red colored parts are the lysosomes. Lysosomes can destroy. They have this lytic enzyme which can degrade this nanoparticle. So in the case of healthy cell, we can see there is a decline in the the nanoparticle is being dissolved. But in the case of cancerous cell, there is an agglomeration which we can see. So this agglomeration is seen in the case of cancerous cell. So endocytosis, as explained, is a cellular process that involves the cells absorbing the external materials by engulfing them into the cell membranes. This diagram is also represented in this form. We can see the nanoparticle clearance. So this is cleared. This has been cleared here, whereas in this it is being agglomerated. So this causes destabilization in cell death. So nanomaterial toxicity. In the case of metals, it can cause DNA cleavage leading to genotoxicity and mutations. In metal oxide, it causes radical generation and protein denaturation. Carbon-based materials, it can cause tissue remodeling and improve fibrogenesis. One of the aspects of nanomaterial is that nanomaterial has vast applications. So it can also be used. It can also be used as a scaffold for tissue regeneration repair, and it can also be used as an organ. It can be also used as an organ by using these metal and metal oxide particles or fabrication methods. This carbon-based fullerenes, carbon nanotubes, diesel exhaust. This is having mostly the mechanical and the electronic devices approach, and quantum dots like the metal chalconite that causes toxicity effect of reactive oxygen species generation, liquid peroxidation, and DNA damage. Then we have the silica particles that interfere with the blood platelet formation, vascular endothelial clotting abnormalities. And dendrimers and lipids, which are heavily studied for drug designing, they cause membrane damage and denaturation of proteins and the lysosomal damage. So I want to conclude my presentation by telling that This nanoinformatics, it contributes to the standardization of data and descriptors, aiding in regulatory compliance. Drug design helps to ensure responsible development and use of nanodrugs. These models are used to predict the potential risk associated with the exposure of nanodrugs and facilitate early stage decision making.